Yeah, can you? Now it is audible? Excellent. Okay. Is it audible, Gogras? Okay, good, good. Okay. Take your time. Right, okay. Hello, Gogras. Can I speak? Okay, great. Thanks. Yeah. Can you all hear me? Sorry for lot of confusion and, working with this technology. First of all, I thank Dr. Balraj Singh, the Honorable Vice Chancellor of Rajasthan Agriculture University. And then I have another, my favorite Vice Chancellor there, Dr. Janaki Ram from YSR University, that is from my place. And he is doing great job there. And then I see there are several distinguished scientists. I enjoyed lectures through this YouTube. And I really thank organizers for giving me this opportunity. Microphone testing. Hello. Yes. Hello. Yes. Can you hear me? Can you hear me? Yes, I can.
Yes,
Can you
I'm here.
Okay, okay.
Yes.
Thank you, sir.
Your voice is clear.
So my
Okay,
voice
okay,
is
thank you.
clear. Is it audible for everyone? Someone says yes. Someone please say yes that it is audible.
Saurabh. Yes, sir. Yes, voice is on, sir.
Oh, voice is on. Thank you very much. Okay. So, well, thank you again for the opportunity and lot of confusion. And I'm sorry to keep you waiting. It is already probably sleep time for a lot of kids there. However, my topic is on gummy stem blight resistance in watermelon. And we are from West Virginia State University. My research institute is called Gassar Douglas Institute. Looks like it's to move. Yeah. So this is gist of the city we are living. This is called Charleston, West Virginia. And this is a beautiful campus, beautiful city. So that is about from where I am, but it is very cold. And I really miss Jaipur climate and the Jaipur food. Had I known that this conference is happening in the Jaipur, probably I would have showed up there. Just to talk about, can you see this cursor? Here is the DNA, that is 2 nanometers DNA. Once it is wrapped around these nucleosomes, then it becomes a histone fiber. Here it is 11 nanometers, and then that is coalesced into much more complex fashion into coiled coils, and finally large size chromosomes. So the genes are embedded inside these chromosomes. And then we have to entangle it and of course sequence it, or can collect tissues from the various samples and do the transcriptomics. For the genomics and epigenetics, we have to do massive sequencing. And also for proteomes, we have LCM, the mass spec and all that can characterize proteins, but we don't do much proteins in our laboratory, but we do a lot of metabolomics. So the integration, the phenotype that we are trying to understand in the watermelon, that is gummy stem blight, we need to sequence various populations to understand the location of the genes that are causing resistance to gummy stem blight. And also the transcriptomics is taking the tissues and studying how they are expressing, etc. But we work on many crops. We work on pumpkins, pomegranate, peppers, melons, including bitter melons, lot of things. Our research is not only on the resistant genes, but nutraceuticals, fruit quality, health effects, etc. So this is kind of population that came to the United States probably in 15th or 16th century in the form of heirlooms. When the slaves were coming to the United States, they brought these heirlooms with them. And then these are highly unimproved, and then they don't have sufficient sucrose content. They are not very attractive. But this is the founder population using which the United States breeders developed the watermelons here. And also we use grafting. One of the presenter was explaining how important is grafting for growing tomatoes in Gujarat. Here also in the Europe and United States, lots of fusarium wilt in the soil. That is a big hindrance for growing cucurbit crops. So we always graft on the lacenaria. There are robotic systems that can graft 10,000 seedlings in a kind of 10 minutes or so. So large size plantations can be supported with the robotic driven grafting technology. And also we do a lot of molecular genetics to understand seedless traits. This is a diploid watermelon, the small flower and smaller leaf. And then the fruit is same size like diploid and tetraploid. So for making seedless, first we have to convert this diploid into tetraploid. And then the tetraploid is large size flower, large size leaf, large size stem with a lot of trichomes in it. But the fruit is exactly almost same size. So we are doing a lot of genomic studies to understand why the fruit is same size. So these are the cultivated watermelons today in the United States. Okay, these are bred in our place by a USDA geneticist, Dr. Amnon Levy. And the biggest challenge in the watermelon is gummy stem blight. So gummy stem blight is caused by three cryptic staganosporosis species, Citrulli, Cucurbitaceum, and Caricae, and all of them collectively they manifest the symptoms. That's why it is very difficult to control. So we made a mapping population. What is a mapping population? The wild types are highly resistant. So these are all Citrullus amorus, which is a wild species of watermelon that cannot be edible and it is almost bitter. So this Citrullus amorus types are moderately resistant to gummy stem blight. I wouldn't say highly resistant. So to pool all the genes together, we made intermatings. And then 4 cycles of intercrossing was done. And then without any selection of these progenies, we mated them with the cultivars. Charleston Gray is one of the iconic watermelon cultivar in the United States. And all of them are highly susceptible to gummy stem blight. So these were crossed again back with this progeny randomly, and 4 cycles of intermating was there. And then 7 cycles of self-pollination is there. So this kind of strategy, intermating various wild and cultivated species, is to introgress the segments like here. there are, say, different types here, different varieties, A, B, C, D, E, F, G, H. And first you have to intermate A and B, C and D, E and F, G and H, and then further intermate like A, B, C, D within this nested matings. So this nested matings will create populations that have introgressed with the wild segments sufficiently and then fixed in the form of recombinant inbred lines. So this is the population that we used for mapping the gummy stem blight. So to start with, first we made evaluations in the field and greenhouse, how this responses of gummy stem blight in this introgressed recombinant inbred lines. And then here is bulks. This is highly resistant and these are highly susceptible. That's why the lesion size is larger here from 5.5 to further up to 10. And whereas resistance is just 2.5 to 2.5, I mean 3.5. So here you can see the range in these box plots. All the rills manifest resistance in this fashion, but resistance are here and susceptible are here. So there is quite contrasting response for the resistance to the gummy stem blight in these bulks. So these bulks constitute 30 different individuals, mixing equimolar concentration of DNA in a bulk, and similarly equimolar concentrations of DNA from the susceptible rills, 30 of them, and making them the bulk. So once we have these bulks, we sequenced in the NovaSeq genome, and then these reads are mapped to the reference genomes. So the watermelon is sequenced in our lab, and Amarus is sequenced in one of the collaborators' lab, Amarus, the wild species. So we used both the genomes. And then once we have this, we performed this one analysis that will tell us where are the important genes, and then identifying those positions and converting them into assays so that we can use these assays for screening larger segregating populations or germplasm resources. So the R bulk, once we bulked all this resistant bulk, then we got quite a bit of, 563x coverage, so deeply sequenced. And then the raw read is 139 million raw reads. And then all of them are very high quality, and 99% of them will map to the genomes. Similarly, the susceptible bulk, when this is the genome of wild species, that is the Citrullus amarus, and this is the genome of Charleston gray, which is the cultivated genome. So the reads that we got from the R bulk and S bulk were separately mapped to these genomes, and then 99% is the mapping ratio. That's what I am trying to say. So all the sequences get mapped to both susceptible parent and the resistant standard sequence, in similar fashion. So this study gave us a lot of variance, especially when mapped with the wild species genome. So it's almost like a 7 million variants, 6 million SNPs resistance, and similarly 6 million SNPs in the susceptible, and similarly 1 million for the resistant ones, and then about 500,000, 500,000 or so are the SNPs to the susceptible genome. So this clearly tells us, if even just looking at this data, you can see the lots and lots of polymorphisms when we mapped with the wild species, as well as the resistant bulk has more SNPs, even it is mapped to the cultivated genome in comparison with this susceptible one, indicating that most of the SNP locations came from the wild species, got introgressed into the cultivated species. So this is another way of looking at, like all the SNPs that we have in the bulk, how they are kind of strongly associated. It calculates almost like a LD within the bulks. And these are the SNP distribution. SNP distribution is the number of SNPs that are there mapped to various chromosomes. Here on chromosome 3, you have large number of SNPs. almost 125, 12,500 SNPs here. So you can see here with the USBL, there is large number of SNPs comparatively to the Charleston Gray, which has a lesser number of SNPs comparatively, indicating that the introgression happened in the rails. That's why they are showing lots and lots of SNPs with the Citrullus amorus. And then this is a analysis that shows are there any really resistant loci for these genomes? So here, when we used the USBL 246 and performed this QTL-seq analysis, wherever you see this peak touching this blue line, which is 99% confidence interval. So if all these locations have the SNPs or the genes that have highly resistance, for the gummy stem blight. Whereas even the wild species has some susceptible ones at 95 intervals. But whereas Charleston Gray genome indicates all of them are susceptible. Resistance is towards negative. The delta SNP index is negative, and then the susceptible ones are towards positive. So you can see so many susceptible alleles are kind of pooled in this cultivated watermelon genome. And similarly, there is another analysis that identifies more QTLs that is called G prime analysis that pretty much uses LD, linkage disequilibrium within the bulks. So we have identified lots and lots of genes and SNPs that are strongly associated with the trait that is gummy stem blight. And most of these genes are known genes for the disease resistance, several of them. And then some of them are matching with the previously published results from different laboratory. That indicates, wow, our data is good enough, because they are in fact retrieving all the genes that are previously identified as well. So we have identified lots of genes and lot of variants. Some of them are just SNPs, and some of them are even PAVs. Presence and absence kind of variation, or insertion deletions with containing the large sequences comparatively to the SNPs. So one home-taking message is 213 genes are there in the Citrullus amorus in the wild species that are not there in the Charleston Gray. Okay, and then 295 genes are there in the cultivated species that are not there in the amorus, the wild genome. But one thing we need to understand here, none of these unique genes are important for the QTLs. So the genes that are shared between the wild species and the Lanatus, that is the cultivated genome, alone have all these homologs for disease resistance. So we did Jivas using these magic rails. These rails are called magic rails. Once you intermated with the wild species and Lanatus and self them for long time, these are called magic rails. And we have about 298 rails. And this sequencing resulted about 1,068 variants, 68 million variants. Yeah, it's a lot of data. And that gave us quite a bit of SNPs when mapped to the USBL genome and Charleston Gray genome. So here, the RILs that are of them, three groups. One is, you can say, susceptible group and moderately resistant group or resistant group. This we can say that is USBL group, that is the wild type group. And then this is the totally cultivated group, and this is the introgressed group. So within the introgressed group, we need to look for the moderately resistant or highly resistant types. So these RILs were subjected to disease resistance, disease inoculum at 7 days, 14 days, 21 days, 28 days, and field. and then in the greenhouse as well. So they were thoroughly tested. And then this Jiva shows clear signals across all the days. They have very common associated SNPs, as you see here. And then most of the SNPs are showing association at multiple days, 7 days, 14 days, 21 days, 28 days. They are kind of causative SNPs showing resistance or susceptibility. And then you can compare here, 7 days, 14 days, 21 days. Here on chromosome 10, we have a gene, and that SNP is showing up all the way across. And some of them, you see this particular SNP is showed 7 days, 14 days, but it is absent in the 21 days. And then at 21 days, we have totally different genes. So it looks like gummy stem blight resistance is quite dynamic, and it kind of alters once in 7 days, 14 days, 21 days. Different candidates, different gene candidates are important for being this resistance. 7 days, you see, here there is no SNP, but here is strong association at 14 days. And this association is there even at 21 days. So, and these two genes, they are associated only at 21 days. They were not previously associated at 7 days and 14 days. So, folks, identifying a resistant gene is not a big deal. And we need to understand how that resistance is held throughout the crop duration. And then, especially on the chromosome 10, the most important marker that has the highest signals of association. And then that one has great allelic effects, especially at 14 days. This is the resistant allele, and this is the susceptible allele that is showing up very susceptibility. So here is the resistance. And in greenhouse also it is resistance. 21 days also it is resistance. AUDPC curve also it shows association. And the field level also it is highly associated. So with all this exercise, after spending $1 million, what did we get? Some of the genes that are showing resistance across the duration, at least, especially this one. Several of the genes, their resistance is very stage specific. At the 7 days or 14 days or 21 days, or in combinations. But this is the only SNP that is consistently associated with the trait. And here is again on chromosome number 1 that is showing association at average field rate. At the field level, this one is showing very strong association. So this is very important. So using this data for plant breeders, we can predict risks that can be used in the breeding program. In the sense, these models will predict the reals that have all the favorable genes, all the favorable in the sense, all the resistant alleles are pooled in this population, in these predicted reals. So you can see this reel performance is quite good, even at 14 days, at 7 days, and 21 days, as well as average field rate. They are all showing very strong resistance in comparatively to susceptible types. So these predicted reels will be immense important to release as pre-breeding lines so that the plant breeders can use and introgress for this important disease. And similarly, here is another predicted set of reels for the AUDPC, etc. And then we have huge collection in the United States, though in 15th century, the American breeders got just a few lines, but eventually we collected the entire world germplasm in our gene banks here. So we collected about 1,500 Lanatus types, all from all over the world, including India, Turkey, Africa, several places. And then we subjected them for the resistance. And then how the resistance is distributed in this population that we obtained from all over the world. And then we used this field level data of this 1,500 lines to estimate, to identify some of the SNPs. And then some of the SNPs that we previously identified in the magic reels are showing up here in this germplasm analysis as well. So we can understand one important gene is very well known resistant gene is a coil domain containing protein, which is very important. So this one, here is a networking of all these varieties, global varieties that we have collected. So here is the susceptible varieties, and then this is the moderately resistance, and this dark green is highly resistance. So how these mutations will happen? This undergone 2 different mutations, and then the population will break up. The resistance and susceptibility kind of alters based on the mutation dynamics that this networking is showing us. This is called haplotype network. These models will tell us how disease is kind of selected in the population when we are doing inadvertently plant breeding without worrying about resistance. Here you see the Tajimas D is 2.2. That indicates population geneticists will say that it is highly positive selection undergoing lot of mutation. So the genes that give resistance to the cultivars undergo lot of mutations comparatively to the other genes, comparatively to the normal genes. So this is for similar characterization of another important gene that is on the chromosome 10, the most important gene. So the initial population probably undergone several mutations, and then these kind of phenotypes happen. And sometimes resistance is broken, and sometimes are built up because of this kind of positive mutations happening, having this Tajimas D 2.8. So we converted most of these markers into PACE assays so that these PACE assays can be used. If you want to do in India, the gummy stem blight resistance breeding, you don't have to do all the exercise what we did. You can just take these PACE assays. They are very cheap, probably $50 each. That helps you to screen thousands of individuals like this. This group, blue group is resistant and red group is susceptible, and these of them controls. So similarly, we have used different important markers and converted them into these PACE assays so that the plant breeders can use it for their selection. So all these are called allele discrimination plots that can be readily used in the field level. You know, you can bring samples and identify whether the line that you have developed is a resistant or susceptible, or the segregating lines can be subjected to select for gummy stem blight even without performing any inoculation, or genomic selection can be practiced. So all these things are going in our laboratory, doing this QTL-seq, all these candidates. Some of this, the most importantly, there are deleterious alleles, folks. So when the plant breeders are breeding, they inadvertently selected a lot of susceptible alleles, and the recombination breeding cannot separate them. So I heard many speakers in this audience also, they were talking about genome editing. Of course, we are all doing genome editing to purge out these deleterious alleles, to clean out these deleterious alleles from the cultivars, not only in the watermelon, in all vegetable and fruit crops. This is an important endeavor. So this is funded by USDA, and we have collaborators in ARS, Amna and Levi, and the North Carolina State University is a strong collaborator. Todd recently retired. And then we have a cook cap, a kind of CAP project, in the sense coordinated agriculture project. Just like in India, we have ECRIPS that coordinate and do genomic resources and testing collaboratively. So these are some of our students, and that is me standing there probably 7 or 8 years back. All the students are now moved out except 1 or 2. Thank you very much for your patience. If any questions, I can answer. Probably Gogras, the scientist, great scientist from IRI, who has visited United States for 3 months, but made enormous contacts across here. And he is in touch with me, so he will probably convey me the questions if you have anything. So let me open other one
Thank
also.
you, Professor Reddy, for making a very informative presentation on a very, very important aspect. And I have
Dr.
only one
Balras,
query.
you are doing excellent work. Gogras told me many good things about you, and you control 30 different universities and guide many young people and scientists. That is very great. And I wish I see you in person someday. And similarly, Dr. Janaki Ram is a great vice chancellor and very positive and really building up a lot of research infrastructure in Andhra Pradesh. So you both are in fact stars of horticulture in India.
So, Dr. Reddy, I have one question that this gummy stem blight now we are facing in case of bottle gourd across the country. But this problem is quite large in state of Rajasthan.
Wow.
And Ranjan, Dr. J.K. Ranjan, he is working at IRI, he is principal scientist. So what you would like to suggest him, what he should do, whatever you are doing in this watermelon?
Yeah, in fact, this bottle gourd, the Lazenaria is sequenced and a lot of germplasm collection is there. So this kind of analysis should be performed in bottle gourd. And sometimes these markers what we have identified in watermelon are kind of useful for other cucurbit crops also. Especially gummy stem blight is a devastating disease, not only for Lazenaria and also cucumbers and melons. So the watermelon is the most susceptible. So we can use these resources across because most of the genes and genomic regions are conserved. So these are highly useful, Dr. Singh. Yeah.
So thank you, Professor Reddy. Anybody, any question from the audience side? Any question? If there is no question, so Professor Dr. Jankiram, you would like to ask? Okay, yes.
Students can email me also. We have a lot of opportunities to work in our laboratory, and I am kind of graduate coordinator here. And I prefer to take our students, Indian students, as grad students. So if anybody has good GRE and TOEFL, their assistantship is sealed here.
Thank you very much, Dr. Umesh. Nice to hear again your presentation. And thanks to you, special thanks for providing opportunity to one of our student recently to work in our laboratory.
That's right. I have students from YSR University.
Yeah. Thank you very much, Umesh. Bye.
Yeah, thank you. Bye.
Okay, thank you, Professor Umesh Reddy, for making a very informative presentation. Thank you very much.
Thank you, sir. I miss seeing all of you in person.
Future with your institution.
Someday I will visit. Please visit us. So, Vice Chancellors, please visit us. We will host you.
Thank you.
And scientists also can come for any short trainings. Students can join us, grad students. And thank you all. And I really like that Jaipur thali. I miss here. And I'm jealous of you all that you all had great food. And
So now, friends, we are inviting the second speaker, Dr. M. Ali Zeda from Iran, and he will be speaking on a very different topic, that is introduction to wild fruit trees in northern Iran with special reference to wild pomegranates. So, Dr. M. Ali Zeda, welcome. If you are हाँ उसको बंद करिए हाँ उनको करिए ज्वाइन कराइए